Hi, my name is Katie Pearson at California Polytechnic State University, and I'm excited to talk to you about some phenological research about the California poppy. Phenology is the study of the timing of life history events such as flowering or fruiting in angiosperms. And phenology is really important not only to the plants, but to the ecosystems in which plants are found, because the timing of growth and reproduction can determine the fitness of the plant itself in a number of ways, from pollination success to dispersal of seeds. And then the phenological events, or timing thereof, can impact other creatures with which the plant interacts, so their success due to the availability of food, habitat, or other resources. Phenological events in plants are often cued by climate variables, and these relationships with climate are often very taxon-specific. So we have been looking at a lot of different species in the community to see how they respond to climate, and this is especially important in the face of climate change. So the questions that we looked at in this research was, how do long-term temperature and precipitation affect flowering date? Then we wanted to understand how sensitive flowering date was to interannual variation in temperature and precipitation. And then we wanted to know whether the sensitivity to interannual variation depends on long-term climate conditions. So does the long-term warm or coolness of an area determine how that species responds to climate changes during that year? And we were studying these questions within the iconic species, the California poppy, Eschscholzia californica. Before I go into the methods, I wanted to talk briefly about what drove this research. This research was empowered by the work of the California Phenology Network. I am the project manager of this network. It is a National Science Foundation grant that funded 28 California and West Coast herbaria to digitize their herbarium specimens. Together, we aim to image, transcribe the label, georeference, and score the phenological status of over 1 million herbarium specimens. And so these are pressed, dried plant specimens stored in these herbaria. And not only are we capturing the geographic and types data on the label, but we are also scoring the phenological status of these specimens by indicating the presence or absence of reproductive features. So we are in year 3 of this network's progress, and we have accomplished quite a lot. And particularly, we've been able to phenologically score over 1 million. We're at 1.4 million herbarium specimens worth of phenological data. So this is a really exciting data set that we can now use to study the phenological events and how they are changing in the California flora and beyond. All of the data that we have collected are all stored in the CCH2, or Consortium of California Herbaria 2 portal. And the data are publicly available, and this is the data that I'm also going to be talking about and using in our research. So our methods were relatively straightforward. First, we scored the phenological status of herbarium specimens based on the presence or absence of reproductive features. Then we cleaned the data set and associated climate data. And then we did multiple regressions to determine the relationship between climate variables and the phenological status. So we scored the phenological status of herbarium specimens of the California poppy using a number of new tools that are available in CCH2. In general, we were able to score herbarium specimens for the presence and absence of flowers by looking at the images individually and then scoring for the presence or absence of those structures. We were also able to score many specimens using text fields. So we have a new text mining tool also in CCH2. In this list, you'll see a list of text fields, and we were able to assign certain phenological scorings to those specimens to which those text fields belong based on the text that we could see in those labels. Then we downloaded those herbarium specimen labels. This is just a screenshot of a CCH2 search. So in the end, we had a little bit over 4,000, but that was before data cleaning steps. The phenological data are stored in a separate data file than the main specimen records, but we have a script that we use to combine the phenological data with the specimen data, and that R script is shared publicly at this address. We then cleaned the data set and associated climate data. We just removed specimens that did not have flowers present, and then we also removed specimens that had missing data, so of course date, coordinates, and phenological data. The climate data that we used for our analyses were from the PRISM climate group. For each specimen, we calculated the normal value, so this is the mean 120-year conditions in the month and the location of collection. And then we also calculated the anomaly, so this is the difference between the normal value at the location and the value during the month of the collection of location. So anomaly can be summarized as how much warmer or cooler than average it is at that location. And this is of particular interest because it may indicate the plant's ability to respond to climate change of the future. Now for our linear regression analyses. In general, our linear regression models included climate variables as the predictors and the flowering date in terms of day of year of collection as the response variables. And when we explored the parameter space to see which seasonal variables were most influential on flowering date, we found that for temperature, the season of April and May was the most influential on flowering date. And then for precipitation, we had two windows, February and March, and October and November. And those were both influential on flowering date in our final models. So first question, how do long-term temperature and precipitation affect flowering date? And so this is asking a question of the effect of climate normals on flowering date. We found that flowering date became later with higher values of long-term mean annual precipitation. And so flowering date was later at wetter sites, you could say, and that flowering date is earlier at warmer sites. Then we asked how sensitive is flowering date to interannual variation in temperature and precipitation. And so this is examining the effect of climate anomalies. So how much warmer or wetter than average that year is, how does that affect the timing of flowering for California poppy? So what we found is that flowering date was delayed with increased fall precipitation, but it was advanced with increasing spring precipitation. So depending on when the precipitation comes, it can either delay or advance the flowering time. Last question was, does the sensitivity to interannual variation depend on long-term climate conditions? And so that is, does the sensitivity in that year depend on how, on average, warm or cool or wet or dry that area is? So what we found is that flowering date was advanced with warmer springs in general, but the magnitude of the advancement depended on the long-term climate conditions at the site. So in cooler locations, California poppy was more sensitive to changes in temperature. And then in warmer locations, there was very little change. Here in this graph, the red bar are warmer locations, and the slope of this line is much less steep. And so in warmer years, they're not flowering much earlier than in cooler years. In comparison to in cool areas, so cool long-term average areas, they're responding much more drastically in warmer years compared to in cooler years. And we did not see this same pattern in precipitation. We only saw an effect on sensitivity of temperature to long-term mean annual temperature. So what could this all mean? Well, one interesting fact about these results is that they're consistent with a lot of studies. We've seen that long-term temperature is associated with advanced flowering date, and then precipitation is associated with later flowering dates. We've also seen many studies that have shown that flowering date is advanced with increased temperature in that year of collection. However, very few studies have been able to tease out the relationship between sensitivity to temperature and long-term temperature at that site of collection. This indicates that there is some intraspecific variation along the range of the species. And considering the latitudinal and longitudinal differences in temperature across the range of the California poppy, this could be pretty influential for the species. So the implications of this finding is that estimates of phenological sensitivity derived from single locations might not accurately predict sensitivities across a range, and that could be a key different factor when you're trying to determine how the plant is going to respond to climate change. This is also a good example of how patterns of intraspecific variation can vary between taxa, particularly because other researchers have found opposite trends, so opposite sensitivity gradients with warming and with precipitation. And there's also a potential that this lower sensitivity in warmer climates for this species could reflect developmental limits or evolutionary constraints. So this could mean that the poppy cannot shift its flowering date anymore in these warmer regions, which could drive a shift of the range of Eschscholzia californica into cooler areas if early flowering is required for the plant to grow and reproduce. Finally, our results show how digitized phenologically scored herbarium specimens can be a powerful tool for understanding the sensitivity of different taxa to climate, especially in the face of climate warming. And particularly because we're digitizing so many specimens and making their data available, we can now study intraspecific variation in sensitivity to climate because our dataset size is big enough. So that's a really exciting result and a good prognosis for using CCH2 data and other phenologically scored herbarium specimens in the future. And with that, I want to thank my team and thank you for watching. Please email me if you want more information and keep your eyes open for the paper which will be coming out in Madronio hopefully this year. Thank you.